So welcome back this afternoon to the next panel. I think over the last day and a half, or I suppose it's actually a day, we've heard really how the reinvention of the entire biomedical enterprise is going to require a transformation in the way that we understand data, collect data, and understand the evidence for what we do. And at the center of this, I think, is going to be a revolutionized clinical trial system. We have to hold on to the core of what we need to make decisions and choices about new data, but we have to do it in a much more efficient and much better way. So to lead an expert panel discussing global clinical trials, next generation design and scalability, it's my pleasure to introduce Dr. Marc Sabatine. Marc and I were co-fellows together at MGH, and he is now the chairman of the TIMI study group, one of the largest clinical research enterprises in the world. He's the Lewis Dexter Distinguished Chair in Cardiovascular Medicine, and he really has been a leader in clinical trials, and his organization has been a leader in thinking through the next generation of these trials. Welcome, Marc, to the stage, and the panel also.
Great. Well, fabulous. Let me echo Callum's welcome to all of you for joining us for this panel. Clinical trials are certainly near and dear to my heart. Dr. Brunwald was and is my mentor for the clinical trial aspect for the TIMI study group. And so TIMI has been doing clinical trials for now over 30 years. But as Callum said, all of us in the field have started to look at clinical trials and think, how can we do them better? These trials are quite large. In some ways, I would say from the bias perspective of a cardiologist, that in cardiology we enjoy perhaps the highest evidentiary standard for our trials. Right? We have the largest trials, hard clinical outcomes. That's what attracted me to cardiology as a resident at Mass General. All that and having Roman DeSanctis as a role model and mentor for cardiology there. But the evidence base for that was amazing when these trials came out, and I said I wanted to be part of that. And that certainly continues today. But if we think about the trials, in some ways we are a victim of our own success. Right? We've set the bar for hard cardiovascular outcomes, cardiovascular death, myocardial infarction, and stroke. As our care has gotten better, those event rates have gone down, and that necessitates trials with that paradigm that are then very large. And whereas TIMI 1 was a couple of hundred patients, the more recent trials that we and the other groups do are routinely more than 10,000 patients, 20,000, sometimes even 30,000 patients. And to pull off that enterprise requires typically gathering 1,000 sites at 30, 40, 50 countries around the world, going through all the regulatory burdens for that, training the sites, having the sites find the patients, and go through the necessary study procedures for acquisition of data. Then putting together that package and placing it at the altar of the FDA and other regulators for review, and then to our peers as well for their judgment. And so with that daunting size and those costs, it's really become quite prohibitive. And with limited financial resources, then the question is, are we hurting ourselves? Can we not have enough investigation into different fields rather than putting in an unduly amount of money for large individual trials? And that's the challenge that we all face. I'll admit right up front that I don't have the answers to any of that, but luckily I'm joined by an outstanding panel who's thought about these issues and how we can tackle them. I will underscore, as may have happened in other panels, we'd like your questions. So I have an iPad device here that when your questions come in, I'll see them and I will try to integrate them into the 45 minutes or so we have for discussion. Let me start by introducing the panel. Going further to the left from me, first Elisabeth Björk, who's the Vice President of the Cardiovascular and Metabolic area at AstraZeneca. Then to her left, Dan Bloomfield, who's the Vice President for Cardiovascular, Metabolic, and Women's Health at Merck. To his left, Jonathan Plehn, who's the Vice President of Cardiovascular Medicine at Covance. To his left, Norm Stockbridge, the Director of the Division of Cardiovascular and Renal at the FDA. And then to his left, Scott Wasserman, the Vice President of Cardiovascular and Metabolic at Amgen. So thank all of you for joining us here today to share your wisdom. We have already chatted a bit in advance and thought about some of the questions. And so I want to go through these and have some structure for it. So the first thing I want to talk about are the outcomes. So, I think back, and I show this slide, thinking back to TIMI 1, where the outcome was TIMI flow grade, right? And now it's cardiovascular death, MI, or stroke, and sometimes in some trials even all-cause mortality. Thoughts about the outcomes we should be looking at. And as I go through these questions, what I'd like you to try to put an additional spin on is, does it depend where you are in the development of the compound? Because there's a lot about sort of pragmatic trials and simplifying stuff that may or may not be applicable at different stages of the development of a compound. So maybe Elisabeth, if you want to start us off.
Very happy to do that. And first of all, thank you, Marc, for inviting me to Boston and to this very good session. I really do think you have to start with the patient population. And what matters in what outcome to look for is going to be important in different patient populations. I think looking at the hard outcomes is going to continue to be very, very important. But as we go to sort of maybe more pragmatic designs, then maybe the all-cause mortality could be an alternative or something to really look at.
Yeah. Scott, we've talked about this topic a lot. As we look at these outcomes, I guess a couple thoughts. One, we've done work together for lipid lowering where LDL was a reasonable surrogate to start with. What about other compounds in the pipeline where there isn't LDL cholesterol as a reasonable surrogate? And for the definitive outcomes trial, what are the outcomes in your mind that we should be thinking about now versus sort of the legacy outcomes we've inherited from trials in the past?
Yes. So when I reflect on this, I think it boils down to kind of the scientific principles. And I credit one of my mentors, and I've been privileged to have lots of mentors in my life. But when I was in Michael Gimbroni's lab as a medical student, he was very keen on making sure you had a very clear hypothesis. And so when we approach clinical trial development and endpoint selection, similar to Elisabeth, there are two kind of real key questions. So what's your hypothesis, and how are you going to test it? And so obviously, I made this joke the other day, but we're not going to test an anti-prostate cancer drug in women. Similarly, we're not going to test a postmenopausal osteoporosis drug in men. So you need to have your population match the endpoints that you're looking for. So I think it's really important to have a clear hypothesis, understand your molecule and know what your molecule does, and then think about how you can design endpoints such that they match or that your modality has the highest likelihood of being able to move that endpoint in a meaningful way for patients.
Yeah, thank you. So Norm, let me ask you, you guys face perhaps a tsunami of requests coming in for surrogate outcomes and have the unenviable task of trying to judge what would be reasonable. What are your thoughts as you've seen this field evolve over so many decades?
So, we do get a fair number of requests to consider novel biomarkers as surrogates. And in some places, I think we've been receptive to it. There are relatively clear areas where you can relate the mechanism of the drug to an outcome and proceed with reasonable assurance that you're going to get where you need to. There's an intermediate stage where there's a reasonably likely outcome. Something constitutes a reasonably likely surrogate. And we have the flexibility of making a decision based on such an endpoint, and then requiring the demonstration of the effect on the real clinical outcome as a post-marketing requirement. But cardiovascular medicine is difficult because it has been really a graveyard of programs that have failed to realize their expected benefits because of various reasons. There are all kinds of inotropic drugs, mostly in heart failure, that have not lived up to their expectations. And so in those kinds of areas, we're sort of stuck without good confidence in knowing what the relationship between a change in a biomarker is and the change in the outcome. You end up having to ask for the outcomes.
Yeah, no, I think your comments are spot on. I mean, I think all of us trained in cardiovascular medicine, remember well the story about PVC suppression, right? I mean, that's sort of the classic one drilled into us for a training, that you worry about PVCs and ventricular arrhythmias. And wouldn't it be good to suppress it? But it turned out in the trials for that it actually increased mortality. I think the other area, actually part of the endocrine world, but certainly related to cardiovascular, is for diabetes. And there has been much debate for, I think many people here will know, the requirement to show cardiovascular safety for diabetes drugs based on observations for some older compounds. And there were a series of trials that were then done to meet this regulatory requirement that many of them showed a neutral effect on cardiovascular outcomes. And some people started to question whether it was worth it putting in all that money into those trials. That being said, we do now have several trials that have shown a very large benefit. And I think ultimately that's a credit to the FDA. If we hadn't done those large trials, these would have been compounds all listed on the basis of hemoglobin A1C lowering. And the benefits for cardiovascular outcomes wouldn't have been as well defined. So I do think there are large advantages. So if ultimately at some stage we do need to have the outcomes there, then how do we construct this enterprise in terms of gathering the data? Now, there are some trials out there for approved drugs looking at different doses, but at the notion of perhaps even bypassing the site and going straight to the patient. So Dan, let me ask you, could you ever see a model for that, right, where we sort of kind of direct to consumer, if you will, direct to subject interfacing there, right? When would that come up in, different for aspirin doses? When does that come up for a compound that's going to be in your armamentarium?
It's a great question. I think it's early days, but allowing clinical trialists to interact directly with patients would be a radical change. And I think what's radical about it is it changes the definition of an investigator. Mm-hmm. Because now you're not going to have 1,000 investigative sites. You can have as many investigative sites as there are patients out in the world that want to participate when that's possible. So that's appealing, for example, in rare diseases where there are very few patients around and you want to reach out to them. In big diseases, outcome studies in cardiovascular diabetes, I think it's also feasible that you could take that approach, but there are many questions you want to answer, not just to inform consent up front, but study conduct. Are they taking their medicines? How are they being cared for? Are they getting standard of care? And then eliciting adverse events and outcomes. So I don't think you can go directly to patients and assume you can do a trial between clinical trials and the patient. You're going to have an investigator in there. It's just a question of how to make that work.
Yeah, no, I think that's right. Again, there's a lot of talk about those approaches, and to Dan's point, I think it makes great sense to try to understand them and harness them. But as Elisabeth has brought up in prior conversations, the life cycle part matters tremendously. and a certain stage, you would need an investigator. Elizabeth, your thoughts?
Very happy to comment on that. Because I think we need to start to think differently around when in the life cycle the study is being conducted. It's very different if you have a new medicine, maybe a new mode of action, and maybe trying to explore the efficacy and safety in a patient population that is at high risk. Then the standard randomized clinical trial, where you need a lot of data, both on efficacy and safety, is very, very important. That's very different compared to when you have a drug where you know the profile, you know the safety profile, and maybe you want to explore it in a primary prevention setting or in a broader setting. And that's when I see that we can really make use of registries and also capturing the data the way we are discussing here. So I think we really have to be much more different in how we are looking at the different trials depending on where you are in the life cycle.
Yeah. Jonathan, let me ask you, so as we think about sort of reaching out to sites around the world or maybe bypassing sites depending on the stage, what do you view in terms of actualizing that? What do you view as some of the issues we'll need to grapple with?
Well, I think there's got to be a more efficient way of finding the right sites. It's not just reaching out to sites, but it's finding sites that have been productive in the past and that are rigorous in their approaches. I harp back to the Tomcat study that we've talked about in this group and also yesterday in the heart failure trials, that when they had trouble recruiting Tomcats, they went to Eastern Europe, where everybody goes to get increased recruitment, and they went to the Soviet Union and Georgia, Russia and Georgia, actually. And they had some problems with some of the patients that were recruited. And if you take out those two countries, the Tomcats NIH-funded trial was a positive trial. The outcomes were positive, and it really was because of the design of the recruitment that was the problem. So what we've tried to do at Covance, which is a contract research organization, which was purchased by LabCorp, which is a basically drug sample analysis company that many of you have probably used or have heard about. We tried to combine our resources to find the sites that are most appropriate, that between patients who have certain blood test abnormalities, we can look for where those are focused and go to the physicians that seem to have that sort of patient, and that's in the United States right now. And also we use our clinical laboratory, which is the largest in the world. We do about 50% of the blood analyses for industry-sponsored trials, and we can look at sites by looking at the lab kits that have gone in and out to see which ones have been productive. About 20% of sites on average don't recruit at all in these large outcomes trials. So to try to eliminate that 20% and to make sure that the ones that do recruit don't have a lot of problems, is really critical. And when you go to areas of the world that you may not have worked in that much before, it's harder and harder to do. So we've been using our lab system to get data-driven results for clinical sites so that we know that they're reliable when we go to pick them up.
Yeah, no, I think finding the right sites is something that we all think about and grapple with, and you laid it out very, very nicely. Norm, let me ask you, from the FDA perspective, the tension between the global trial, in part to get global representation, but as was appropriately noted, the costs are vastly different around the world. The trials are already extremely expensive, and extremely expensive particularly in the United States to do. But the FDA is looking after, narrowly if you will, the interests of patients in the United States. So how do you as an agency try to grapple with those two opposing forces?
Yeah, so clearly we would like to promote drug development in the United States, in part because it ensures that the people and care that is given is applicable to the United States. But fundamentally, we do not insist that trials or development programs enroll patients in the United States. What we require is that some belief, I guess I'd say, that the results achieved wherever they were are applicable to the United States. That usually means standard of care that's similar, diagnostic standards that are similar, and where that's been achieved, we've been perfectly happy to interpret the results and provide labeling. I think the most dramatic case of that, I would say, is the approval of Plavix post-MI was based on a 40,000 patient study done all in rural Chinese hospitals. So there isn't an insistence on drug development being done in the United States, but we want what you want, which is to know that when you employ the drugs, the results that are in the label are likely to be what you're going to see in your clinic.
Yeah, no, and I think that's right. I mean, obviously the cost advantages are obvious, and the question really is there effect modification based on the practice of care there, so that a benefit or harm seen in a certain area would be different in the United States because of or more westernized practice because of the facilities we have. One of the themes that came up was this notion of finding not only the right sites but the right patients. Maybe just to touch briefly on the notion for genetics, and maybe Scott, for you to comment from Amgen's perspective with its acquisition of Decode for how that will inform us maybe for drug development, and does that help in the development stage? And I'm thinking for PCSK9 inhibition as an example there, where the validation of the target in the genetic studies also, I think, gave confidence during the development that the results would pan out, which thankfully they have.
Yeah, I mean, we've certainly tried to leverage our core competencies in genetics to discover new molecules and get more confidence in terms of the benefits and potential risks of therapies that we're developing. And so, not dissimilar to what Elisabeth was saying, it has to do a little bit with your courage of your conviction, but also the timing. And so, when we were developing our PCSK9 antibody, Repatha, we looked at Decode and leveraged their capabilities to determine both what we thought would be the benefits, but then also the potential risks. And so in terms of kind of looking for signals and so on, there was questions about neurocognitive issues, in which we went to their data and they weren't able to find it because they have a very well phenotyped population, an elderly population with a lot of kind of good cognitive markers, et cetera. And so we've looked at it very, very carefully, and I think it can really help you focus your development efforts, both in terms of efficacy endpoints, but then also safety evaluations going forward.
Yep. So let me talk a little bit about the 21st Century Care Act, and amidst all of that, there was a comment about real-world evidence, and I think it's worth spending a minute or two parsing that a little more clearly in terms of the notion of how do we gather the data? Is it through the infrastructure of a clinical trial with all the bells and whistles that are expensive bells and whistles that go with that, versus observational data? And I'd like to just sort of go down the row and get people's opinion for how they view those opportunities in terms of the need to randomize versus the need to use or being comfortable with registry data where the fidelity of that data may not be verified with the same rigor that we've done. So Elisabeth, why don't you start us off?
Yes, and I think the use of real-world evidence data, real-world data, is going to be increasingly important as we move forward. And I think there are two different parts that we need to think around here. First is the observational data, which to me is that you look at the efficacy and safety once a drug is on the market, and you can then explore that drug or that class of drugs or a certain treatment modality, and you do that within the label that you have. And you use registries, etc., to get that information. So that's one part. The other one is the concept of prospective randomized registry trials that I really think that we should try to discuss here, which is that you identify your patient population through a registry, you randomize your treatment into that, and you collect the data, the information, using registries or electronic healthcare records. And you do not use any standard CRF, so you make use of the data you have there. And you could then go outside the current label and do that instead of a randomized clinical trial. And in my world then, that's what you can do when you know your drug and when you have done maybe one or two of the randomized clinical trials, and you have a better sense for how the drug works and what the safety profile is.
Yes.
But if
So
you could
at a
do that
later stage.
stage,
Yeah.
but if you can do that, then you could get very important information in a different patient population to a much lower cost.
Yeah. Dan, your thoughts?
Yeah, I agree with what Elisabeth just pointed out. I'll just maybe highlight a couple of additional things. One is there may be two reasons to do a real-world evidence trial. One would be because you're concerned, or the community is concerned, that the standard of care in your trial isn't representative of the standard of care out in the real world. And so if you want to understand how your drug does in the real world, that's a way to do it, to have as light a touch as you can on the way in which medicine is practiced. That's one potential value. I think that's what Elisabeth was alluding to. A second is, I think, more down the road is how can we begin to use real-world evidence or pragmatic trials, maybe that's a better term, to enhance drug development and improve the way in which we innovate. And so if you can do studies, let's say as Rob Cale likes to talk about with cluster randomization, where you can randomize within a healthcare system 200,000 patients, you can get a lot of really important data. You can't get non-serious AE data, right? You can't get really precise measurements of blood pressure or things on exam. But if what you care about is hard outcomes, you can get hard outcomes from the medical record. And I think we'll have to go through a phase where agencies look at the data that comes forward from these kinds of data sets and has to feel comfortable that they trust the way in which data is analyzed and how it's brought forward. If you can get to that place, then I think it would really spur innovation, because the cost of those trials would go down, the speed would go faster, and you'd be able to make a larger claim down the road. One other caveat I'll just add, and I'll stop, is that's more possible when you have marketed drugs. As soon as you get into investigational drugs, now there's issues around drug supply, drug storage. It gets much more complicated than that. So I think where we are today is, for marketed drugs, I think that's a real potential path forward.
Good. Jonathan?
I think it depends on what the question is, what the hypothesis is that you're testing. And there's really a nice editorial in JAMA last year by Hal Sachs, who was my former chief of medicine. And he talks about internal versus external validity. And the kind of real-world trial that you're talking about has great external validity. It looks, the inclusion criteria are pretty wide. It's valid in all sorts of populations, but it may not answer the question that you really want to ask early on in development of the drug. And that really needs to be tight data. As a company, we oversee a lot of these trials, and we look at all of the protocol deviations and the transcription errors and all sorts of problems that can mess up your phase 2A trial. And therefore, that kind of trial is really, it's inappropriate, I think, to do a wider range trial, because you really don't need to answer the question about who it's applicable at that level to, but whether the drug's mechanism of action works. So your data has to be really, really tight. You have a small number of patients, and you want to maintain your statistical power by reducing variability. But as you move on, then you can sort of morph into the real-world setting.
Yeah. Norm, I'm going to pass you for a second. I want Scott to comment, and then we're going to get the regulatory perspective at the end of this line of questions.
Yes, so I'll paint a little bit of I agree wholeheartedly with what everyone else has said, but I want to kind of paint a forward-looking picture, which is we're in a very unique time in terms of the practice of medicine. I think John Leichleiter was up here and he was talking about the biocentury, which is clearly where we are with genetics and the evolving informatics platforms. But it's also like biocentury meets Moore's law, because we have an amazing ability right now to potentially aggregate lots of different data sources, curate them, standardize data fields, such that we can do real-world clinical trials with investigational therapies. Now, we need to, crawl before we toddle, before we walk, before we run. But I do think that, we need as a community, both academics, industry, and regulators, to really think about this going forward. Because I think, we live right now in a time where there are not endless resources to do these clinical trials. The economies and there's lots of financial pressures on everybody, and we have to prioritize. And I think there's an amazing opportunity there if we just seize it. But I think it's going to take all of us working together to find a solution there.
So before I ask Norm to comment, a few thoughts that I'll add, and then I'd like to hear very much what Norm has to say. I think for the issues, I think randomization is key. I think the observational data are hypothesis generating. But from our own bias, seeing hormone replacement therapy, vitamin C, vitamin E, all these things that looked very good in observational studies with careful multivariable adjustment, did not pan out in the randomized trials. Now, it's interesting. We had a bunch of folks from Google who were visiting us at the med school, and that was my second or third slide for a presentation. I had lots of other stuff to talk about, but they said, well, wait a minute. How many variables do they adjust for? And I'm like, well, they adjusted for 20. And they're like, 20 to what power? Like, no, just a total of 20 variables. But so that seems small, to them. But still, I worry that without randomization, it's very hard to tease that out. But once you do randomize, how you gather the data. There are a lot of opportunities, and hearing the notion that the amount of sort of hand-holding might vary depending on the stage, I think makes good sense. Part of the hesitation for that is just thinking, at least in Partners, when we adopted Epic as our electronic healthcare system, one of my best friends who was in the CCU right before me said, be careful if you tick off diabetes, because there's like 4 other screens that come up. And so it's challenging for that. Now, depending on the outcomes you have, then that's not an issue. So Norm, your thoughts for how the agency, thinking ahead, might look at this. And as Scott said, it's not going to be tomorrow, but if we're going to move it forward, where are the areas that you see we have the greatest opportunities now for kind of traction to do things differently?
Well, I do think that there are that the main thing is to preserve the randomization. I think that for the things that you decide you care about and want confidence in, we can talk about various things you can do and the cost savings associated with less structure around the data collection and the resulting problems associated with follow-up and completeness and accuracy. But at the end of the day, for the things that matter, there is no substitute for randomization. And for the things that are regulatory decisions, there are not going to be many circumstances where you should be ready to give that up.
And let me then push a little bit further. So I'm hearing, I think, consensus amongst the panel for the importance for randomization, certainly to establish the efficacy of the drug and to make sure that it's safe as well, to make sure that the two groups are balanced, with non-randomized data being complementary to that and hypothesis generating. in the data gathering, that's such a huge expense for it, right? How do we push that? How do we push that forward? Thinking ahead, could you imagine a case where the data that comes in is just whatever is gathered through the electronic health record, right? Just to push that point a little bit further. Let's say they've been randomized in some cluster randomization at sites or practices, and then you want to know for death, MI, or stroke, and you just extract what's happened in the record, and it is what it is. And adverse events, I mean, they're not going to be routinely captured. Where do you see that ever fitting in terms of approval and the sort of life cycle, if you will, of a compound, in terms of the first time you're looking at it or for an additional indication?
Well, it certainly is wildly different than the way we behave now, right? I mean, we evolve these ridiculously complicated definitions of clinical endpoints. How many pages does it take to describe what's an MI? And then we don't believe an investigator, even if he's a cardiologist, can determine what an MI is. That takes a committee. It requires adjudication. Really? I think we can take numerous steps back short of relying on the electronic health care records to do things more efficiently without much compromise on the quality of the data one is achieving.
And I guess let me add an additional point to that, maybe for other people to comment on as well, and then we'll give you a breather, Norm. That so you've always been incredibly thoughtful and forward-looking and quite practical, as you said. Why can't a cardiologist sort of determine what's happened to his patient? That's what we do in clinical practice. And yet, when the trials are done, it's not just your opinion, but there's the Office for Scientific Investigation looking, and all of us have sort of gone through this, where like a IRS audit, not that I've had one and I'm not asking for one, but the notion of the dotting the I's and crossing the T's for stuff, if you have trials that have tens of thousands of patients, potentially thousands of outcomes, where we know, of course, not every MI is going to be reported. That's why we randomize people. And yet there's this notion of I have to follow a certain packet through and make sure that it went the proper way. Maybe to have the sponsors and Jonathan from the CRO perspective just sort of go down and talk about within your own group, how do you grapple with sort of sage forward-looking advice you get, and then your regulatory group saying, but at the end of the day, we have to make sure that it's all going to be squeaky clean and unassailable.
Well, sponsors demand that right now. Most sponsors do for big trials like that. Even in phase 2 trials, we're getting requests to have an endpoint committee. I've sat on a lot of endpoint committees before I got into the CRO world, and they're sort of useful, but at the end of the day, when you actually look at the differences and the changes that are made in terms of the diagnosis, they're really very few. We actually did an analysis of that in the Busendelol evaluation survival trial, BEST trial, heart failure trial, and it really didn't change things very much. So I don't think there's been a lot of research into this, but I think it could be made much easier. But in pragmatic trials, there's even been talk about patient-reported outcomes. And I think that's going a little bit too far because patients don't really understand their disease processes very often.
No, I think that's right. I mean, I worry, the patients who we see in practice, their notion of when they went into the hospital, whether they had a heart attack or not, or whether they were admitted for a rule-out of a heart attack, that is not obvious to people. Elisabeth, your thoughts?
I would be the first one to agree that I don't think adjudication is adding much value, and it's actually very much driving costs. So I would be very positive to run pragmatic clinical trials with the clinical endpoint being sort of what the physicians, but not the patient, has reported. I really wanted to push Norm a little bit more on this. It's one thing to design a pragmatic clinical trial and be very specific on the endpoints that you are looking for and just capture that. But what about then taking the next step and get that data from a registry or from sort of healthcare records or something like that? Of course, with the notion that we are randomizing patients into this and all of the above, but just the data collection. What are your thoughts around that?
So, I think there is a fair amount of openness to trial designs that involve non-classical ways of collecting the information. Yes, it's true that the inspectors will go through a classic site and look for problems. But what fundamentally undermines the process is bias, not noise. And if we're still talking about randomization and blinding, and you've got a mechanism that has pretty good follow-up, the kind you can get with a conventional trial, you can probably tolerate a fair amount of noise if it's cheap enough to collect the information that you want through some mechanism. And it fundamentally does not matter to me if I can't do 100% source validation of a trial. It matters to me if things hang in the balance of, 1% one way or another, and you've got 12% missing information. And maybe the trial's not well blinded. There are circumstances where it's going to be hard to interpret if things are sort of close to the wire. But, there's no fundamental reason why you have to be sure you captured every MI.
Dan, your thoughts?
Yeah, I completely agree. It's about bias, and you see a result, and you can ask the question, if you looked at the data more carefully, or you did another survey of events, would you uncover data with such a bias that it would change the result? And that's, I think, what Norman's getting to. It's just very unlikely when you see a robust response. And so, yes, you need a robust response, but once you have that, there should be a different level of scrutiny of the data and the sites on things that aren't likely to impact that result. And I think one of the challenges is, and Norman alluded to this, is the field force at the FDA, they have their mission, they go out and do their thing. But what they do affects the ecosystem between the sponsor, the investigator, and the agency. It costs a lot of money. So we have to get aligned about what we want investigators to do, and then get alignment that the agency is comfortable saying, okay, you're not going to collect that data, and that's okay.
Scott?
Yeah, I'll just again kind of throw out a provocateur kind of comment. I do think that as we evolve our statistical methods and our understanding of data and randomness and data and so on and so forth, I do think that some of this will be machine learning, artificial intelligence, where you can identify the outliers just based on the pattern of the data. And so some of these things that Norm's alluding to, which I agree with, I think people won't be doing those anymore. And we'll just have an ability to have someone look at the data and say, we need to go look at this site because there's something funny going on here. So I think the work that we do now is going to change over time. And I think, our job is to kind of look forward and try to plan for that.
Yeah. Well, good. I'm glad we've solved all the issues here. But I think in the remaining 1 or 2 minutes we have, let me try to sum up. It's been a wonderful discussion, and I think it's indicative of the whole forum here, bringing together people who interact all the time but view the question through slightly different prisms is incredibly helpful. I think the themes that I've heard that have emerged are, first, the fundamental principles are always important. what is the scientific question we're asking? What do we think this molecule will do in whom? That needs to be set up extremely clearly. What are the right outcomes to look at? Randomization being key for that. And then in terms of the process, again, going back to the fundamental principles, our goal is to eliminate bias, right, to have confidence in the data in terms of minimizing missing data, but not in terms of looking at small questions for data completeness or verifying individual pages of documents. That's not going to make or break a trial of 30,000 individuals. And so then it's going to be the question, I think, for the next generation of trials to start, getting traction and grabbing some of those concepts and saying, for this next trial, we need to get agreement up front. As Dan pointed out, we can all agree here, and if it was just up to us, we'd be set, but it's not. And so there has to be a clear plan for that so that one group can then be successful in achieving that, and then that paves the way. And so I hope with the discussion we've had here today, we've outlined some of the steps for cardiovascular trials that we can do that, and hopefully in future years we will have achieved some of those successes and continue to progress in the field. So that's the end of our time. I want to thank everyone for spending this hour with us. Thank you.